Not all berries are fruits, but all fruits are berries. Let's enjoy a bit of learning about phylogeny, or how things are nested together in an orderly sense. So for this objective, for this lecture, your objectives are to define descent with modification, branch, node, clade, most recent common ancestor, species, fossil, vestigial structures, and homology, as well as analogy. Draw and label a phylogenetic tree. Be able to describe the case of Louisiana versus Schmidt. Give examples of phylogenies supported by fossil evidence and differentiate between microevolution and macroevolution. So, descent with modification. This is one of those terms of evolution we discussed in an earlier lecture. Oops. There you go. If you look at the dog breeds, you're going to ask what types are most closely related. Now in this example, I just say the gray wolf and then just a whole bunch of other things. So we have a source species that we know of in our historical sense, which was the gray wolf. Now we also have all these other species which are, or species? No, they're individuals. Okay, well, can a toy poodle mate with a Great Dane or a greyhound? Well, no, but they're still the same species, right? Well, are they? This is kind of a job for phylogeneticists to determine whether to split a group or to keep a group together. So descent with modification is kind of a classic idea for evolution. So how a group of organisms descends from a common ancestor. Okay, let's kind of look at that. Remember that evolution, in its stricter sense, is descending along these trains with modifications as things go on, depending on what current selective pressures exist. Evolution is not goal-oriented. Now let's take a look at this step by step. I'm going to say that we are each a population of organisms, and we're all going to start out with the same general instruction, but we're going to adapt in different ways. And here's how we're going to do it. I want you to start today's drawing exercise with a circle. Now, next, I want you to draw two smaller circles. Now, fill in two of your three circles. And then I want you to draw a semicircle. And finish all that off by drawing a triangle. Now you may have your own answer, and what's going to happen is at each step, people have different solutions to different problems. Just like a population that is put in different areas is going to come up with different solutions to even the same problem. So how many of your circles looked like mine? You have a circle, two smaller circles, two circles filled in, a semicircle, and a triangle. If yours looks different, that's because although the selective pressures were the same, the solutions were different. And this is where descent with modification can kind of come along with having different solutions to common problems, something we're going to wrap up with at the end of this lecture. And this is really this kind of step-by-step idea. So that phylogenies are going to evolve along certain ways. So we have, your common group. So this is our most recent common ancestor. And at each of these steps, you're going to have a branch originating from a node. So we have our most recent common ancestor. We all started with a circle. Now, some of us are going to end up with that. Some of us are going to end up, something a little more abstract. Realistically, at each step, where I said draw two smaller circles, fill them in, draw a semicircle, draw a triangle, we're going to end up with different patterns. And realistically, each one of these subsets is going to branch off as well. Now, not all subsets are going to be followed. Some of them will go extinct, such that we might actually have something like Mickey Mouse here. Mike Wazowski? I don't know. But we're going to have a whole bunch of different steps at each one of these. Our most recent common ancestor is the same, but at each time we get a node, we're going to have a branch. Now, we could include all of them that have, circles containing the whole answer. There you go. Are going to be a clade. And maybe this would be the clade of everything is still touching and making something that looks like a face in general. That's a clade, is a group of organisms close to each other. So these are your different terms. Now, you might want to review in Bio 142, we covered phylogenies as well. So this would be a good time to go back and look at how phylogenies are created, because we're going to be looking at a lot of phylogenies. All right, so one of the ideas of phylogenies, of course, is that all you can do is modify existing states. And this is within a clade, all things are modified from most recent common ancestor, which is going to give your basal state from which things can derive. So remember that when we're going through these, that each of these clades is merely modifications from the most recent common ancestor of that clade. Let's look at some how we apply this phylogeny idea to a couple case studies. Involving HIV. So over time, in a patient, HIV evolves. Different strains are going to evolve within a single human immune system, such that the end strains are different from, but derived from, the original. Now, one of those strains is going to get passed on to another individual, so that different strains are present in a population of humans. Patient 1 may have gotten this light blue strain, and a whole bunch evolved in her, and then patient number 2 got a light greenish blue cyan strain, and a whole bunch of different ones evolved in him. And patient 3 got this orange strain, and a whole bunch of different ones evolved in her. So you can actually trace phylogeny of HIV between people. The most recent common ancestor for each one of these clades within a person is going to be the infection, the one that infected them. So you can trace the infections between individuals by looking at phylogenies. And this is what was done in the case of Louisiana versus Schmidt. SCHM. Awkward. Okay, Schmidt was a doctor. He had patients who had HIV. And he also had an ex-lover named Trahan. So Schmidt was accused of infecting Trahan with a patient's HIV. And the suspicion was really that Schmidt had come over to Trahan's house, and Schmidt had a special injection for Trahan. They thought it was a medication, but it may have actually been blood of Schmidt's patient. So Trahan accused Schmidt of giving them HIV. Well, we have two hypotheses. Hypothesis B is that Schmidt's patient's HIV is completely unrelated to Trahan's HIV. This is what Schmidt would have said. Schmidt says, I did not infect Trahan with HIV. Trahan got HIV from another local group, in which case Trahan's HIV would be more similar to the local controls and would have a most recent common ancestor that is not similar to Schmidt's patients. Whereas Trahan said, I got my HIV from Schmidt, their patient. In this case, Schmidt's patients and Schmidt's patient's HIV and Trahan's HIV would be very similar. The results here are Trahan, you see the two strains of Trahan's HIV, showed more similarity to several strains of Schmidt's patient's HIV than to local controls, thus confirming hypothesis C, that Trahan got their HIV from Schmidt injecting Trahan with HIV. Schmidt was then sentenced to 50 years of hard labor starting in 2000, so still going on. Okay, and that's how phylogeny was used. The most recent common ancestor is that Trahan shares more in common with Schmidt's patients than with local controls. Well, where does HIV come from? That's another question that can be solved by phylogeny. So human HIV has more in common with certain chimpanzee HIVs than with other simian viruses. So we got that one from the sooty mangabe and the stump-tailed macaque. So you have some types of HIV look more like other simian immunodeficiency viruses, but most are going to be coming from the chimpanzees. And this is because we believe humans got these viruses from bushmeat. So they went out and killed a few monkeys and got blood on them, and that blood infected them. This could also be because people were trading monkeys as pets, and you get monkey bites, you got monkey blood transmission. That's going to lead to certain amounts of HIV moving between organisms. So the most recent common ancestor shows that group M here made the switch to humans actually in the 1940s, many years before it was actually detected and described, more like in the 1980s. Okay, so there's some case studies where phylogeny was used to confirm where HIV came from and who's giving HIV to who. Let's kind of discuss some evidences of evolution as well. So start off here. Evidences for evolution. It's a bit of a switch over here, but we'll move back to phylogenies in the end. So we're looking at different reasons that we might actually say that evolution is reliable. So now that we've covered what evolution is, descent with modification or change in allele frequency in a population over generations, let's look for cases of it that we can easily grasp. First off would be microevolution. Microevolution is a great evidence of evolution because it is evolution, just on a smaller scale. So Garland in 2003 actually did natural selection for mice that had a high basal metabolism, the HBMR, or a low basal metabolism, the LBMR. And what he was looking at was mice running on a wheel. Certain mice ran on a wheel more often than others. So he would actually be studying, he took the mice that ran on the wheel the most and crossed them with other mice that ran on the wheel the most, and found that two populations could diverge from a single population. What was your residual BMR minus the standard deviation adjusted for body weight? And you see that over time these actually diverge. So this is microevolution, changes in allele frequencies in a population over generations. These high runner mice actually ended up resembling humans with ADHD. They even tried giving Ritalin to the mice, which made them actually slow down the amount of running they had. And I think these mice also had anxiety. So it's interesting to see how genes are similar to each other across different mammalian species. Another good evidence for evolution, and indeed a good evidence for phylogenies, are vestigial structures. Useless or rudimentary parts of an animal, like whale legs. So whale legs, there isn't selective pressure to have or not have these structures. Now, I was actually told by a creationist that whale legs are all the same, and that there is great selective pressure for whale legs, and that without the legs, the whales cannot live. This would not explain why some whales have no legs and some whales have stumpy legs coming out of them. So the leg bones are absent in some and present long enough in others that they actually stick out just a little bit. So that seems to be a pretty good contradiction, but a very testable one. Do whales that lack the bones have a higher or lower fitness than whales that don't? So is there actual selection against or for? It's a testable hypothesis. All you have to do is measure a whole bunch of whale reproduction and then dissect the carcasses. Okay, it's not a practical hypothesis, but it's testable. You can't say just, well, it doesn't have anything. You can test to see if it does or not. So whales do have variation. This variation, is it heritable? Studies haven't been done yet. Does it cause differential survival and reproduction? Studies haven't been done yet. But what they have been done on is sticklebacks. So the amount of armor that sticklebacks have and these spines in the back of their, on the dorsal side, these vary based on how much predation the sticklebacks are under. And you'll actually see that some will have much less armor, and there's no selective benefit to having the armor, such that even there might be a selective benefit to having less body armor or less spines. And these are vestigial structures in the species of stickleback that harken back to when the stickleback actually needed these things. Perhaps a better example of vestigial structures that's a little more direct to you is your vitamin C gene. Yes, you have a gene that has the ability to make vitamin C. All apes do. Now, in some monkeys, this gene is quite active, and the monkey is able to make vitamin C on their own. However, in you, oh great ape, you have the gene, but the gene is broken. Well, why would you have the gene at all if you only got it broken. So this vestigial structure is evidence of a past time when a population of monkeys were able to make their own vitamin C. Now there's no fitness cost to having a broken enzyme if you can get enough vitamin C in your diet, like a high fruit diet. So you should be able to make your own vitamin C, but you didn't need to, so there's no selective pressure against not being able to. And you still have the gene, it just doesn't function. Which really raises a kind of question for the alternate hypothesis, which is humans are perfect organisms. Well, why do they have a broken gene that doesn't work? Well, there's some reason. No, you gotta test it. You gotta test these things. It's also present in guinea pigs. Guinea pigs have a broken gene for vitamin C as well. Now, it doesn't mean we're related to guinea pigs because, most fascinatingly, theirs is broken in a different way, showing that one of their ancestors lost the ability to make vitamin C. So these vestigial structures, you thought I was going to go for appendix or tonsils. Nonsense, we have plenty of uses for those. No, your broken vitamin C gene. There is no use for that. Then we have evidences of speciation. Given different diets, you can have some fruit flies, and you can feed them on starch or maltose. And over time, they will become adapted to living on starch or living on maltose, and they'll actually begin to show mating preferences to other flies that feed on starch or feed on maltose. Now, it's not a complete speciation event because some still can mate, and if you just inseminate these flies artificially, they'll still produce viable offspring. There's also a hybridization event between two different species of flower in the American Southwest, producing a new sunflower species. So we can see divergence or fusion of species. Well, maybe they aren't species. Well, maybe a Chihuahua and a Great Dane are the same species, even though they can't reproduce and even though their offspring are not viable. Maybe they're still the same species. Maybe they are not. We see this as kind of messy. We see hybridization between relatively unrelated species like Bos taurus, the common cow, and bison, the bison. Should have been a guinea. Those can actually hybridize to make beefalo. So that hybridization ability or the ability to split like this really makes species messy. So stickleback species, we see this pretty often with divergence of benthic versus limnetic sticklebacks. So you'll have a stickleback species introduced to a pond, and eventually over time there'll be a benthic species that lives on the bottom of the pond, and a limnetic species that lives around the surface of the pond. This happens repeatedly. Almost every time a stickleback is introduced to a big lake, it evolves into two different populations. Now the benthic species share a most recent common ancestor with the limnetic species, not with other benthic species. This shows that the most recent common ancestor diverged over time, using a phylogeny to show the repeated divergence of benthic and limnetic stickleback species. So we know this. Warblers, we have Siberian warblers and Tibetan warblers. We also have a gradient in between. Each species mates with the next nearest species, but if you were to take a Siberian warbler and a Tibetan warbler, they're not going to produce a viable fertile offspring. So this would show how they can each mate with the next one, but they can't mate end to end. So it's a messy species concept. Are they all one species? Like a Chihuahua could mate with a Pomeranian could mate with a Basset Hound with low standards, could mate with a Labrador with lower standards, a Labrador could mate with a Great Dane. You have that gradient in between them where they share genetic information through populations, much in the same way as the Siberian and Tibetan warblers. Species are messy, so we have evidence of speciation is that species are messy. We can't define them well because some are still in the process of divergence. So when is it really a new species? Well, this biological species concept is that organisms can mate, and produce viable offspring. We can't test this for extinct organisms. We don't know if a Stegosaurus and an Allosaurus could mate. Probably not. They look very different, but we can't prove it. It's relatively useless for asexual organisms because asexual organisms don't mate. There are alternative species concepts. We may discuss those in class, or if you have a question about those, pop it up in the forum. But another option is baramins. Messy species, you won't see this on the actual side notes, but orderly baramins. A barramin is basically described as everything that is a species. And it's described as such kind of very post hoc. So horses, zebras, donkeys, and, what else were there? Horses, zebras, donkeys, yeah, anything in between that looks like a horse. It's all one barramin, because those are all the same created type. So that's easily sorted. The problem you have there is it's very roughshod. So you have things like wildebeest and Cape buffalo, American buffalo, bison, aurochs. They're all the same species. Are they? There's really no way of testing it. And not only that, this whole idea of barramins, the created kinds, was only invented in the 1940s. So if evolution was true, we expect to see messy species. If we expect to order them perfectly and easily sort them and easily name them, and they look very distinct, well, that would be really running counter to evolution. So, lions, tigers, and house cats, are those all the same species? A creationist would say yes. And an evolutionary biologist would say no. And it's hard to test. Can you mate a cat and a lion? No. But are they the same species? No. Of course not. But some would say they are, and that's kind of your alternative here, but it's much less testable. If evolution were true, there are transitions, hybrids, and of course, over time, change. All right. This also means there were only 2 cats on the ark. So anytime you see an ark with a tiger and a lion, it's a lie. That's not what they actually believe. So, speciation, we just did that. Okay, so fossils. Okay, also, so descent with modification, let's discuss fossils a little bit here. And this is kind of what we would expect. If evolution were true, what we're going to see is we look at 2 things that are kind of alike, like a hippopotamus and a whale. They're kind of alike, well, they're big mammals in the water. Okay, well, do they share a common ancestor? Well, they share a common ancestor at some point, yes, because all mammals have to share a common ancestor. Well, then the question is, can we find the fossils? And would the fossils be where we expect them to be? How long ago were whales on land? Well, we expect to see land-like whales, or whale-like land creatures, well before we see species that looked like they could have been from on land but were in the water, like more like delphinid or seal-like creatures. And that's what we see. We see these, let me get the name right, Indiohyus, this little rodent-like thing. We see that is more similar to Ambulocetus, which means walking whale. You see those far more distant than instead of seeing Indiohyus at the same time that Doraiodon was present. So Doraiodon came after Ambulocetus, came after Indiohyus, came after the most recent common ancestor with the hippopotamus. And you see these transitional forms in a certain orderly manner. Not only that, but if evolution were true, we'd be able to predict where to find the fossils. So we should be able to find a fossil that is, if we see birds and coleophysoids, coleophysoids. My poor child's brain is just like, no, you're mispronouncing your dinosaur names. Coleophysis should be way back in the Triassic. It is. Living birds should be happening now. They are. We should see something in between before we see real birds evolve. So we should see bird fossil, bird fossil, bird fossil, dinosaur-like bird fossil, bird-like dinosaur fossil, dinosaur fossil. We should see them in that order, and that allows us to predict that perhaps a dinosaur-like bird would be somewhere in the early Jurassic, and a bird-like dinosaur would be somewhere. Some of these are going to be in the Jurassic period. We do see things in the Jurassic. We see Archaeopteryx in the Jurassic period where we predict it. But we might be able to really predict where to find these fossils. And there comes Tiktaalik. So we have Panderichthys, which is a lobe-finned fish, and we have Acanthostega, which is a fish-like amphibian. Now, if you were going with the barramins, the Panderichthys, that is a fish. The Acanthostega, that is an amphibian. Boom, we're able to put them in different groups according to barraminology. Now, these are all extinct species, by the way. Now, Tiktaalik is something that was between a fish and an amphibian. So, we'd expect that it has to come somewhere between 360 million years ago and 397 million years ago. Okay, we need to look in rocks from that period. All right, and it has to be, you're not going to find Tiktaalik in the middle of a desert, so we're going to look for it in rocks that would have been marshes, shallow freshwater swamps at that time. Well, Ellesmere Island had marsh-like environment at 375 million years ago, so scientists went there to look for their fish amphibian, and they found it. They predicted where to find it based on where the intermediate should be, and they found it in the environment at the right time. That is the power of evolution. Now, I've seen this argument refuted multiple times by saying no, Tiktaalik is a fish. That doesn't refute the idea that these evolutionary biologists could predict where to find it, go to the right place, and find the right fossil. The alternative argument, which is never brought up, is that, well, Tiktaalik fossils are everywhere and they got lucky. Because Tiktaalik fossils are not everywhere. Because you find the right species in the right strata. You don't find feather-bearing dinosaurs in fossils from the Triassic. You only find them in fossils from the Jurassic on through the Cretaceous. You can predict where to find the fossils in the world, and that is an important concept when it comes to evolution. Extinct species stopped existing at a certain time. This is not a hard concept, but it's a concept that can be used to predict in which strata you will and will not find their fossils. And that brings us to our last little bit in fossils: homology and analogy. The idea of homology is that things that are related are going to have the same structures. So your human, your horse, your cat, your bat, your bird, your whale, your acanthostega are all going to have relatively similar bones for the limbs. Now, a horse walks on its tippy toes, specifically the tip of its middle finger. Horses are flipping you off. It's okay. Horses are better than us, and they know it. Humans, we actually walk on our wrist, pretty much like the equivalent of the hand bones or wrist bones of our feet. Cats are going to walk on their palms, really, and the wrist bones there, and those bones right there in the palms. Bats going to use them to fly, of course. You get the idea. They all use the same bones, but for different things, and that's homology, having the same features because you share a common ancestor. Our common ancestor being Tiktaalik or acanthostega. Same features, common ancestor. That's homology. Analogy is having similar features, different ancestors. So the most recent common ancestor between penguins and sharks, or between penguins and dolphins we'll go with, most recent common ancestor between penguins and dolphins did not have fins. It had something else. It was some sort of land-walking, lizard-like mammal. That's going to be way back in the Permian, is going to be when penguins and dolphins shared the most common ancestor. It didn't have fins. It was a desert-dwelling creature anyway. Shark fins look like penguin fins, look like dolphin fins, but between penguins and dolphins were a whole bunch of things that were land-dwelling and didn't have fins. So this is analogy. There's similar solutions to the same problem. Fins on a submarine are analogous to shark fins. They're for cutting through the water, but they really didn't evolve. So analogy is similar solutions to a common problem not present in the most recent common ancestor. All right, we have something called molecular homology. And this is a good plug for Dr. Fox's occasionally offered class, genomics. If you really want to get molecular homology, you really got to take that course. We'll touch on it maybe once or twice, but it's a treat. If evolution were true, we're going to expect organisms that are morphologically similar by homology should have similar genetic codes for other genes. So you see organisms that look the same, that all have these hands, should have the same kind of genes for hemoglobin. That would be the same kind of idea. And phylogeny should be supported by molecular similarity. So if you looked at phylogeny, the most recent common ancestor between two things that look alike should be supported by having the same genes and genetic structures. And this is what we actually saw with Trahan and Schmidt. We saw that the molecular similarity was present between these two infections of HIV. But here's the problem, and here's kind of a fun one. Phylogeny isn't always supported by molecular similarity, which can lead to some much more interesting bits of evolution. So, Enjoy, and I'll see you in the group chat.